$6 to $8 a sample, and then IDEX is closer to $10 to $12 per sample. One of the little easier to set up, and you could set it up in your bedroom. I've done, culturing and setting it up in a bedroom that I was staying in. The sample processing. You can run the IDEX sealer off of a car battery, so you don't even have to have reliable electricity for some of this. And depending on the the ambient temperature, you could figure out what is needed for incubation. And so IDEX is also nice because the trays are sealed, and so there's kind of less of a biosafety hazard than kind of plates that can accidentally open, and you could be exposed to kind of high concentrations of resistant organisms, whereas the IDEX trays are sealed. So it is a bit more expensive, but it can sometimes be more feasible in certain settings. I think one of the main reasons why we were evaluating it is because there are a lot of studies that are already planning on using IDEX for enumerating E. coli, being able to work within that infrastructure for looking for antibiotic resistant E. coli is just, I think, helpful for just expanding the opportunity for further surveillance.
Yeah, interesting. We've also done some experiments with aquagenics, but of course it's not for Yersinia coli. That's also something, depending on the context, it could be interesting to use. Okay, so maybe before we go to the mentee question, there's Iftita. Bu Iftita was a lecturer in our study program who's raising her hand.
Okay, thank you. Actually, I'm the one who asked about the clinical isolates one. What I'm thinking, if we compare the sequence in environment with the clinical one, I think we can make sure that the one we found in the water, if it's similar to the clinical isolates, that means the pathogen in the water is causing the disease. We can make sure that, or I'm not sure if you about your idea, but I'm just thinking about that. And also the other not question, but the other question that I want to add is about the comparison between IDEX and TBX agar. I want to ask if we want to do the further experiment after we isolate the E. coli. For example, if we want to clarify the gene using PCR, or we want to sequence from the isolates, can we use the culture can we use the culture in the IDEX one? That's one I want to ask.
Yeah, no, that's a great question. And that's actually what my group is working on, a workflow to go from the IDEX and how we can use molecular methods in conjunction with the IDEX method. So it's a great question, and we're working on it. So hopefully ask us again in a 6 months, and hopefully we have some good results to share with you. But your other question is, about the clinical sequences and the environmental, to be able to make causal claims about the pathway. It's a little tricky because it's somewhat difficult to know the direction of the transmission. You could just be finding actually that, it's human wastewater that's contaminating your groundwater sources. And so that's why you see overlap in the genomic sequences of the isolates from your drinking water and your clinical samples. But if you do some temporal sampling, that might be one way to get around that. So if you kind of sample what's in water, sample what's in the clinical or in humans, right? And I've done this actually at the household level, where we go back and we visit households. So we collect, different types of environmental samples, including water. We've also collected soil samples and food samples. And then we also collected human stool samples at the household. And we would go back every couple of weeks and collect the stool sample and the environmental sample. And so then we can actually see if we see certain strains show up in the environmental samples before it shows up in the human sample, then we can kind of see that maybe causal direction a bit better. But then if we see it in the human sample before it shows up in the environmental sample, then it might be going in the opposite direction. So I think it can be a little tricky to make a strong causal claim if you're just kind of collecting data at one point in time, but it would be suggestive, right? Yeah.
Thank you.
Thank you, Bu Iftita. So she's currently, I think, in the middle of writing or submitting a proposal on doing like clinical and environmental sampling and surveillance in regards to the tricycle program. So let's hope that it comes through. Okay, we have another question in the Menti, which is, will AMR concern influence technical requirements used for local scale or community-based sanitation infrastructure? I'll maybe try to answer on that first, and John, if you have something, please add. I think, to my knowledge, the findings around that is very limited. I think I know Angela did some work on this, but there is a lot of AMR study on the efficiency of centralized wastewater, I know, and different types of biological treatments, but really nothing on the level of on-site or communal based sanitation as far as what has been published. And I know that's part of the plan or the recommendation for WASH documents by WHO, as well as, I think, coalition of scientists that emphasizes that this is an important part to also research on. Maybe Sean, if you want that, or maybe Angela is away further into it. Unless maybe Sean or Angela.
I was just going to, so I don't know much about, and it's a little unclear how resistance, like these different mechanisms that resistance is acquired while in the environment, while say you have some surface water and there's kind of pharmaceutical contamination in the surface water, would that create pressure on organisms in that water to develop resistance, and at what rate and which types of organisms and under which?
Sean, Angela, heads up, we'll take another group photo a few minutes before the end. So for those who probably aren't prepared yet, you can get ready. But just before that, while you're pampering up, I would just like to announce that this webinar is actually part of a Road to Indonesian WASH Symposium that we are organizing. So this symposium will be